So again, today is just an overview of viruses in general. We'll get into more resolution as we go on through the week. The chapter 6, is going to basically involve the inner makeup of viruses and the different types of viruses. They all have nucleic acid, they all have a protein coat, and then some have envelopes and some do not. We'll talk about how they attach to the cell and the steps that a virus has to go through to get from outside the host to inside the host to replicating and then back outside the host. And then the steps involved to go from the infectious particle that invades to then the nascent new viral particles that then exit and go on to infect other hosts. And then at the very end, we'll get into just some very basics on some other infectious particles that are non-viruses. So prions particularly. There's some other types of viruses called satellite viruses, which they're satellite viruses because they need another type of virus to be infecting the cell for them to be able to enter and infect as well. So they can't do it on their own. Okay, viruses were really one of the last microbes to be discovered because they're so tiny. We can't see them, they're hard to grow. And so it dates back even to the days of Pasteur, who thought that rabies was caused by a living thing smaller than bacteria. So way back in 1884, there was this notion that something must else be out there that we cannot culture and see with our microscopes, because clearly these people are being infected by something. It's an infectious disease. They went through the Koch postulates, and we can't seem to find a bacterial cell. So it must be something smaller. And then Dmitry Ivanovsky was the one who later on came up with the term virus, which is Latin for poison. And then he and Bejerink showed that the very first virus discovered was the infectious agent in tobacco, okay? Because back then, tobacco was a huge deal. And when crops started turning brown and getting spots on them, that was a big agricultural and economic disaster. And so these guys started studying what could be causing these tobacco plants to get this mosaic pattern on them of dead plant tissue. And so they found the first virus, and it's called tobacco mosaic virus. And that's going to be a virus that is very well studied because it was the first one found. It wasn't really until the 1950s, though, that virology emerged as a really defined field of study. We had understood it enough by that point to really figure out how these viruses work and the mechanisms that they use. Remember, viruses aren't cellular. They aren't defined as being alive, even though they have biomolecules like proteins and RNA or DNA and lipids from the host. They are not technically alive. That said, each flavor of virus is going to have a specific shape, a specific size, a specific way that it puts itself together. So it's very organized. It's not random and haphazard, okay? We are not sure when viruses came about. It's kind of a chicken and egg thing, okay? When you talk to some people, they'll talk about the RNA world hypothesis, and that is just in a nutshell, that the first biomolecules that really came about were RNA. And then that ruled the planet for a long period of time before that RNA could then assemble and come together with other biomolecules and then be packaged inside a cell. So when do the viruses come out, right? A virus needs to have a host. That's why it's not technically considered alive, because it cannot reproduce on its own using its own machinery. So it must have evolved, some argue, after bacteria and other hosts came about so that the viruses could then infect them. And we'll get into some of the different types of viruses, and one are bacteriophages. So they're viruses that specifically attack bacteria. That is their host. And so people are actually using bacteriophage now as a type of therapy for bacterial infections. If you give the patient a bacteriophage, it won't infect that patient, because viruses, as we will talk about also, have a very specific tissue and species tropism. So a very specific type of tissue and a very specific type of host that it will infect. And so the bacteriophages aren't harmful to the human patient, but they might be harmful enough to, quell that bacterial infection. And as a result, these viruses have shaped the evolution of life by invading and causing the bacterial host, for instance, to respond in some way, changing its genetic makeup over time to protect itself from the virus. And then you have viruses that intercalate their own genetic material into the host's genome, which will obviously, if it's in a coding region, change the structure of the protein that comes off that gene. So viruses have been very, very linked with the evolutionary process of life, for good and for bad. And finally, they're obligate intracellular parasites. So what does that mean? They are obligated to be inside the cell of a particular host. And it's a parasitic relationship. It's not mutualistic. They don't both benefit. Only the virus benefits. Sometimes you could argue that maybe it's commensal, where the virus benefits and the host isn't impacted, but that's really, I haven't seen many cases of that occurring. So viruses are obligate intracellular parasites. They're also tiny, right? That's why Pasteur couldn't associate rabies with anything, because the microscopes back then weren't powerful enough to detect the viruses. We're talking about less than 0.2 microns across in diameter, right? And in fact, in a lab, if you want to purify a virus from an infected cell or host and then use that as a stock to study or to infect other things. What's up? Sorry.
Can I take a break? I've done half of my schoolwork.
Yeah. To use that as we call it a filtrate. So you grind up a bunch of infected cells and you pass it through a filter that has a 0.2 micron pore in it. And so nothing can get through that but the viruses. Okay, and maybe some cellular structures, but you're trapping out all the bacterial cells, you're trapping out all of the host debris. So they're small, right? And they range in terms of their size. So you can have the megavirus, which is 800 nanometers across. That's the biggest. Pox virus is pretty big too, like 400 nanometers. And then it goes all the way down to something like 22 nanometers with yellow fever virus. Okay, not much bigger than a hemoglobin molecule, and that helps bind the oxygen to your blood cells at 15 nanometers. So these are tiny, tiny, tiny things, but there's a dynamic range between the big viruses and the small viruses. Largest viruses, again, megavirus, and then this Pandora virus. So We're talking much, much bigger than most viruses around. Almost on the threshold of detection with a regular microscope, but not quite. Viruses are not cellular, like I said. They don't have the protein synthesizing machinery. They don't have ribosomes, right? To then translate their nucleic acid into proteins. They only have what they need to invade a host cell and control it, to hijack the host's machinery, to bend it to its will and do its bidding, okay? And that is simply, those parts needed are just some kind of covering. So the virus particle itself is a protein coating called a capsid, and all viruses have that, and we'll talk about it in a minute. Inside that capsid is going to be the nucleic acid, either RNA or DNA. And then some viruses, not all viruses, will have an envelope outside of that capsid made up of the host's cell membrane. And then in addition to those major fundamental structures, the capsid, the nucleic acid, and sometimes envelope, there'll be a number of different matrix proteins and enzymes that they use to enter into the cell or to break open the cell, to exit the cell, and also, recruit the cell's machinery to start replicating the viral genome. Any questions so far on this? Okay. They're very geometrically shaped. Okay. And in fact, if you purify them out and they start to, so you have a bunch of viral particles that have all aggregated together, they can even form like a crystalline-like structure. Okay. And we'll talk a little bit about, the anatomy of their capsid and how it can be icosahedral. They look like a very geometric shape, and so they can really line up just like a seed crystal gives rise to more crystals. These viral particles can kind of do the same thing. All viruses, as I said, have capsids, and so that is going to be this structure right here, and this is an icosahedral shaped capsid. The capsid together with the nucleic acid is called the nucleocapsid. So nucleo for nucleic acid and the capsid for the capsid. So put them together, you get the nucleocapsid. Some viruses, as I said, will have an envelope. And that is going to be made up of the host's membrane. It takes it with it. And then embedded into that envelope is going to be certain proteins, the spike proteins. So we've probably all heard about coronavirus and how it has these spike proteins that bind to the ACE2 receptor of the host, right? There's going to be a matrix right here that's in between the envelope and the nucleocapsid. And then there can be other structural proteins as well. So, for instance, influenza, that is a envelope virus, and we get the H and the N from different structures that are embedded into that virus, hemagglutinin and neuraminidase, which we'll talk about later, okay?
Does the envelope always come from a host cell, or can a virus actually create an envelope for itself?
It cannot create its own envelope. Good question. So we'll also talk about how viruses escape the host. And one way to do it is to just blow up the cell, right? And that's an easy way to get out. But you really, if you want to be an efficient virus, when you blow up their cell, you could cause damage that could kill that host, right? And if you can't escape the host in time and get in proximity to another host, the virus replication could stop. And so another way to leave the host cell is to just bud off of the cell. And as it buds out of the cell, it takes with it the membrane of the host. So an envelope is always going to be the host cell material, which is why envelope viruses don't survive outside a host nearly as long as a non-enveloped virus, okay? Because if you think about it, this capsid here is just protein, and protein is pretty stable outside the body with these viruses. But an enveloped virus is using material that is used to being in a nice, sufficiently hydrated 37 degrees Celsius environment, okay? And so when it is thrust out of that, it doesn't survive very long. It has limited time, hours, maybe days, to reach another host before that envelope starts breaking down. Which is a nice thing about coronavirus, is that because it is enveloped, it doesn't hang out outside the body for more than several hours, typically. Good question.
Hey, Carl.
Yes.
So you just said that a COVID doesn't exist for a few hours outside of the host, right?
Well, I have to interrupt you there and be a little bit nerdy. We'll talk about this. COVID is the illness, but the virus, yes, it doesn't survive outside of the host very long.
Then how come when that cruise ship was going on, like in the early days of it, they're saying they found it like on substances up to, like on surfaces in like the cabins, like 2 weeks later?
I don't know.
Because they're saying it lasts really long on metal and stuff. Is there like a reason for that? Or is that just kind of like there was so much information going around that a lot of it probably wasn't accurate?
Yeah, that would be my guess. So, that was at the early days. And there's actually a paper that came out recently that compares the longevity of different coronaviruses. So the initial SARS virus, MERS, now SARS-2, and how they survive on different surfaces. So cloth-like surfaces, metal, plastic, paper, cardboard, and certainly 2 weeks is a very long time.
Yeah.
And I don't know. I didn't hear that report, and it might have just been misrepresented somehow, potentially.
Are we sure the viruses weren't dead?
I think it said living, but I mean, it was also in April, and there's a lot going on, different information being put around. And do the ones without the envelope, the naked ones, do they break down more because like as they're on different surfaces, like the polarity will like break down the molecule? Or
No.
does it...
So non-envelope viruses last a long time.
Okay.
They can last months.
Yeah.
Hava just said that she found a recent study where it lasts up to 72 hours on metal. So,
Okay.
so yeah, and that's kind of that's the longer end of its existence. If you look at it on plastic and other types of surfaces, it can be shorter than that even. And that is because this envelope, again, is made up of components that are used to being inside a body, not outside the body. So not only is the temperature, lower and the hydration is lower, but there's also just enzymes in the environment that can break that stuff down pretty rapidly. Good questions, guys. Okay. Two different types of capsid structures. Helical, where it just kind of looks like a tube almost, and then icosahedral. That's the three-dimensional polygonal shape that looks more like this geometric thing. Okay, and that's this. This is the icosahedral form. It's going to have 20 different sides and 12 evenly spaced corners. Very, very geometric, right? If you look at lots of structures in nature, there is an elegant geometry to it, right? And viruses aren't excluded from that elegance. When we look at helical, they can, so in this lower panel, it's like a rod-like shape, okay? Just a long tube. And that's because the capsid is laid out in a linear fashion and then also tubular, okay? So that's why it's called helical, kind of like DNA. And then that's going to wind around inside the capsid. That is the capsid. That's going to wind around inside the, well, this says naked, but these are clearly proteins. So hemagglutinin spike and neuraminidase spike, those are spikes from an enveloped virus. Let me annotate, sorry guys. So this here, naked virus. This here, enveloped virus. The structure is going to be a little bit different depending on whether or not it's inside that envelope or not. I'm guessing this must be influenza because it has the hemagglutinin spike and the neuraminidase spike, and that's where we get the name H1N1 or H1N5 or H1N9 and so on. It's the type of spike that it comes from, the strain. Sorry, just a second.
How do you get the session name and password for the map testing?
They give it to us
How does she give it to us?
in real time. So he's going to do it at
I know, I want him to do it now. I don't know if
then you got to log back on to Zoom.
Okay.
So anyway, sorry about that, guys. Icosahedral. So that's going to be now. Then we talked about the helical. That's the icosahedral. It's going to be arranged differently, not in that log linear shape. It's going to be arranged of these things called capsomeres. Okay, so capsomeres or capsomers line up in this triangular-like shape. So each little circle, and this is protein, remember. So we have these circular droplets that align and form a triangular structure, okay? And that all together forms what's called a facet. And then the facet is going to be one of those 20 different sides of the icosahedral capsule, okay? Does that make sense? So one of these things here makes up one of these things here. So one of those 20 sides is made up of a bunch of different capsomers coming together, okay? And that's really it. I mean, think of all there are many, many viruses and they infect many, many different types of organisms, but those two structures for the capsid is basically it. Here are some examples of icosahedral viruses without an envelope. So rotavirus is going to be a naked virus. Herpes simplex virus is going to be enveloped. And I don't know if you guys saw the news today, but the Nobel Prize in Medicine was awarded to 3 researchers who had discovered and improved treatment for hepatitis C virus. So that's kind of cool. But anyway, so herpes is going to be.
They had a cure for Hep C now.
Yeah.
They do. Okay, that's what I thought. I thought I heard that on TV. They have like a drug out or something.
Yeah, it's pretty exciting times, and the people studying Hep C virus will, they refer to it as a pandemic because it is across the world, and up until recently there wasn't a cure. So, really exciting, great stuff. It's hard to say if the peace prizes are decided, based on things going on in the world or how they decide from each year. But if the pandemic from COVID-19 influenced that award or not. But anyway, good stuff. I think it was well earned. Here is the herpes simplex virus. And so it's going to have the DNA core, it's going to have the capsid, it's going to have the matrix in between, and then the envelope on the outside. A lot of slides just kind of belaboring the structure, but I want to really bore that in so that you know the different types, because there aren't that many really to focus on. The genetic, what does that say? Sorry, I got something in my way. The general, not genetic, general structure of viruses. So mostly are in animals, but they can also be in plants. Like I had said, the first virus discovered was in the tobacco plant. And so these envelopes that they get are going to be from the host. Most plant cells, they don't have as fluid of a cell membrane, right? They have a cell wall, if you guys have had general biology or botany. And so it doesn't lend itself to budding off as readily as animal viruses, but there are some envelope plant viruses. It acquires the envelope as it leaves the cell. It has proteins on the outside called spikes, and that's what they use to attach to the next host cell.
So the virus would go in naked and it would come out with an envelope?
Sorry, that was very quiet. Could you turn the volume up on your device maybe, or ask again? I couldn't hear that.
So the virus goes in naked and comes out in an envelope?
No, the virus, and we'll go into that soon. The virus has an envelope, and that envelope, once it's left, it acquires the envelope as it leaves. And then that envelope is going to encounter another cell, and it's going to stick to the envelope of the host cell, and then it's going to go inside it. And then the newly formed viral particles are going to leave with a new membrane. So a virus doesn't infect a cell and then that same particle go on to infect another cell. Viruses go in, they disassemble, and then their parts go on to make new baby viruses, and then those go out and infect new things. Okay, and we'll talk about that soon. But that's a good question. It's good to clarify that, what's happening. Viruses can be generally categorized into three major types. One is complex viruses. And so these are going to be things like the pox virus, which is a huge virus, as I mentioned, 400 nanometers across. And then these flexible tailed bacteriophage, this right down here in the second figure is going to be a bacteriophage. And those are the ones that you think about, you know, when they draw these viruses, and, you know, they make them look like little tiny robots. This is a terrible drawing, but you know, they look like this. Like, I gotta come get you, little robots or little insect things. Those are bacteriophages. They don't infect animal hosts, but these are going to be complex in their overall structure. It has this tail. A bacteriophage has a tail, which other viruses don't. And so that's one type. These are going to be considered, here's a much better drawing than the one that I did, but this is a bacteriophage. It's got this capsid on top, and then these little legs coming off, these tail fibers. And don't worry about all this anatomy, like the collar and the sheath and the tail pins and the base plate. I'm not going to ask that on a quiz or on a test, but there might be an image of this and it'll be like, okay, well, what kind of virus is this? And you might need to know that this is what a bacteriophage looks like. Okay. Pox viruses, typically, they tend to lack a true capsid, as we've been talking about, and instead have this thick layer of lipoproteins. So what's a lipoprotein? Anybody know? No.
It's a lipid and a
It's a lipid and a protein, exactly. And remember, they don't make lipids, so this has to come from the host.
What is the structure of HIV? Does it have a lipoprotein membrane?
No, it doesn't. And we'll get into HIV, I guess on Wednesday or Tuesday. But no, it does not have a lipoprotein. Enveloped viruses, that's another type. So we have the complex viruses, we have the enveloped viruses. And again, the capsid can be helical or icosahedral. And it depends on whether or not, if it's helical and it has an envelope, then it's going to kind of like mix around inside that envelope to be packaged, right? If it doesn't have an envelope, oftentimes it's more elongated. Ebola is a nice long helical strand of viral particle. So helical nucleocapsids that are enveloped, think mumps, think rhabdoviruses, icosahedral examples of that would be herpes, HIV. And so they're not going to have the lipoprotein, but they are going to have envelopes, which again is good for things like HIV because it doesn't survive that long on surfaces. Okay, so the fear when HIV first came out, like acquiring it from using a public restroom and sitting on a public toilet seat, was not very, a rational concern because they don't survive that long out of the host because they're enveloped. And then last type is naked, right? And that can be helical. And again, if it's a naked virus, it tends to be more elongated. If it's helical, it doesn't have to coil around to fit inside the envelope. Example being a plum virus, plum pox virus. So that's a pox virus that is a plant virus. And then icosahedral could be things like polio, papilloma. And again, we'll talk about those in a little bit more detail later on. We're going to focus on human pathogens for this course. So when we get into the RNA and the DNA viruses specifically, we're going to focus on those types. It can be, so they're nucleic acid. So what does a virus have? It has protein. It may or may not have an envelope, but it definitely has nucleic acid. And that's going to be either DNA or RNA, but never both. Okay, a virus isn't going to have inside its capsid both DNA strands and RNA strands. And hopefully we all know the difference between DNA and RNA right by now. Deoxyribonucleic acid, ribonucleic acid, their structures are slightly different. DNA goes to RNA, RNA goes to protein, right? That's the central dogma. These DNA or RNA segments are what essentially drives the type of virus it is and what type of proteins it produces and how the structure of those proteins interact with the host. Okay. Some viruses have 2 genes. Some viruses have hundreds of genes. Cytomegalovirus, which is particularly bad for pregnant mothers and their offspring, has hundreds of genes. So it can vary greatly from one virus to the next. DNA viruses are usually going to be double-stranded, but some can be singular-stranded or single-stranded. All right. So most DNA viruses are double-strand. Some can be single. And the way that DNA is structured can make it in a circular ring, or it can be linear. RNA viruses are kind of the opposite. Most RNA viruses are single-strand. However, some can be double-strand, very rarely.
I'm kind of confused because I thought DNA was always double-stranded.
Right? Exactly. I mean, in humans, it is. Until those strands come apart to transcribe off of that genome.
But then technically, isn't it RNA once they do that?
No. So the difference between RNA and DNA isn't just the number of strands. It's actually the chemical composition of nucleic
the U versus T. I remember that. I just isn't it still technically? I guess it wouldn't be because they don't have U, do they? Okay, that makes sense.
Yeah. So for instance, DNA, it's not uracil, right? And it's a very similar nucleic acid, but again, with deoxyribose, it doesn't have oxygen, and ribonucleic acid does. So the molecular structure is slightly different, and that's what makes DNA more compatible and usually in a double strand configuration, and why RNA is usually in a single strand configuration. But in biology, there are exceptions to everything. Anything that anyone ever tells you, there's always going to be something that you can find that goes against that grain. And in this case, it's typically going with the grain, but in some cases there are aberrations. So primarily RNA viruses are going to be single strand. DNA viruses are going to be double strand, but not always. With those RNA viruses, it can be segmented into a bunch of different RNA pieces. So influenza virus, for instance, has multiple different segments of RNA in that capsid. Other RNA viruses may just have one strand of RNA or two strands of RNA. Okay, so it depends on the type of virus. It can be DNA, RNA, it can be segmented, it can be just one segment, one strand. And then to make it even more complicated, it can be positive sense or negative sense RNA. Okay, so single stranded, if you look, I have this abbreviation SS, that just stands for single stranded RNA, which is most of it. If it's positive sense RNA, that means that it can immediately be translated by the host ribosomes into proteins, or immediately copied into other RNA segments. However, if it's a single strand RNA genome that is negative sense, it has to be transcribed into the complementary sequence that then is the positive sense, and then from there translated into proteins. Okay, so negative sense RNA viruses have an additional step that they need to do before they can start setting up their factory and pumping out new viral particles. So besides the structural proteins and the genomic content, the last little bit of the structure of viruses includes enzymes. So some of them have their own polymerases, which a polymerase is used to transcribe the DNA or RNA to make new ones. Replicase to copy the RNA. So again, when the virus enters, it uncoats and then it needs to make a bunch of proteins for the capsid. And then it also needs to make a bunch of copies of RNA to then package into the new particles to then perpetuate its existence, right? That's its progeny. And then some have a reverse transcriptase that we'll talk about a little bit when we go into the mechanism of HIV, for instance, that then takes that takes the RNA segment and synthesizes it in the reverse order, hence reverse transcriptase into DNA. Okay? Any questions on this so far? Pretty straightforward stuff. Okay. So how do we classify viruses? How do we name them? It's not willy-nilly. The coronavirus is named because of its structure. The spikes on it, when you look at an electron microscopy image, it looks like it has kind of a little bit of a halo or the corona of the sun, okay? But in order to name a virus, you have to submit it to this committee, an International Committee on the Taxonomy of Viruses, or ICTV. And my grad school mentor always, conjured up this image of them all being just these crusty old people that are grumpy and don't let you get to name your viruses with fun, cool names. I had mentioned earlier that bacteria, it can get pretty creative in the naming system, but viruses, it's by the book. I've discovered or have helped to discover probably like 7 or 8 different viruses, and none of them are called Franz virus because that would not fly with the ICTV. All right. It has to be based on the structure of the virus, how it's composed, what it's related to. So if you find a new virus, it may not be genetically related to any other virus to a certain extent to make it that same family of virus. So it gets complicated, and I'm not going to make you know the ICTV. It's just there to give you an idea of, when we talk about things like COVID, coronavirus, SARS-CoV-2, that comes from somewhere, right? COVID is not the name of the virus, it's the name of the disease. The virus is called SARS-CoV-2. And that wasn't just what someone came up with. It was very much by the book how they came up with that name. Okay. Distinct viral types share a collection of properties, right? So their host range, what they can infect, the pathogenicity, so what kind of damage they do to the host. And then of course the genetic makeup. So we can sequence their genomes and see what degree of relationship they have to other known viruses. And the more similar their genetic material is, the more closely related they are, presumably. The way the capsid is formed, if it's helical or icosahedral. If it's a nucleic acid strand that is an RNA segment, and the number of those segments, all these things play into the naming system, right? The microscopic appearance. So rhabdovirus, it's called rhabdo because it looks like this rod shape. Coronaviruses, as I just mentioned, looks like the corona of the sun. It can be named on its geographical location or its anatomical location. So adenoviruses are found in your adenoids, right? And so that is why it's called adenovirus. West Nile virus is called West Nile because it was discovered first in the Nile area, right? Eastern equine encephalitis virus, right? I just mentioned, I think last week, that we're seeing increased cases, and that kind of happens every year now in the northern Midwest. Eastern, because it's in the eastern area. Equine, because it originates from horses. And encephalitis, because it inflames your brain, right?
Is West Nile the actual scientific name?
Yeah. Yeah, it is. The West Nile virus. And the naming system, so yes, you can have a Latin version of the name, but West Nile is if you want to refer to it as the species, on that level, it's called West Nile virus. But then if you talk about the family and that kind of thing, where is it like the family, then that will have a different name, a little bit more scientific-y sounding name. Some are a blending of several characteristics, like picornavirus. What do you think of picornavirus? Why it's called that? It doesn't have anything to do with corn. Any guesses? If you pick that apart, what is that telling you about?
I've never seen
Pico
that.
mean like really small,
Yeah.
and then RNA, so it's like a really small RNA virus.
Absolutely, exactly. Picornavirus, pico RNA virus. So, that's where these names are coming from. All right. DNA viruses that infect humans, these are some of the common ones. Pox, herpes, adenoviruses, papilloma, polyoma, Hepadna viridae. What do you think Hepadna is a mixture of?
All I recognize is liver.
Yeah, so HEPA. But actually, I believe the HEPA is a numerical designation, so the number of DNA segments it has. But now I don't remember. Maybe it is liver, right? It could be. But then the DNA, right? Hepadna, HEPA DNA virus. So again, we come up with these naming systems by blending their characteristics. in some cases. RNA viruses, there are a lot of them. Picorna, Calisi, Toga, those are kind of smaller ones. You got flavivirus, those are the hemorrhagic viruses. West Nile is an example of flaviviridae. Bunya. Bunya is another hemorrhagic, but I guess most of them are like dengue fever and stuff like that is a flavivirus. Riovirus, orthomyxo, paramyxo. And we'll talk about these in more detail when we get into RNA viruses, but just giving you a brief overview. Coronavirus, we all know that now. Rhabdo, here it's this rod shape, so rhabdo. Retroviridae, retro means that they have the reverse transcriptase enzyme, and then they go from RNA to DNA.
That arena one, is that RNA?
Yeah. Yes. So this arena viruses, these are all going to be RNA viruses.
I'm sorry, I meant like, is that how they shortened it? Instead of saying RNA viridae, they did arena viridae?
Oh, no, I don't know where arena comes from. It might be, it could have been that that the outbreak was first characterized in an arena, or it could be that the shape of it is kind of like, looks like an arena where you would go see, a sporting event or something. I'm not sure. I'll look that up and let you know, but I don't recall. I don't think it's related to, because it's an RNA virus and they just put an E between the R and the N, but maybe. Okay, so it's 10:50. We are out of lecture time. Unfortunately, it goes by pretty quickly. Let's stop here. We'll take a 10-minute break, and then I'll talk a little bit we'll come up with some ideas of what we can do about the quiz and the Labster, and then talk about the Tuskegee syphilis project. So are there any questions on that material so far? Anything that you were wondering about? And I would like to carve out some time to just talk about viruses and stuff in general, because obviously we are in the midst of a virus and there's lots of questions surrounding that. But
Hey, Carl.
yes, Emily.
Are you gonna finish this same set of this same slide deck on Wednesday?
Yeah, probably. I don't know. Yeah.
I'm just asking for like note taking and stuff, because I am trying to listen to you and take notes. And sometimes for me, I feel like you do like a lot of really good in-depth explanations, and it's hard for me to get the whole what I need to have copied down and actually take notes. So I'm just wondering, because I think I'm going to copy this ahead of time and then actually like listen to you more, I think. So that's I just want to make sure that like this is what I should be copying down ahead of time.
Yeah, I definitely want to cover the stuff that so the next part is going into how the virus multiplies, the stages, and so it will definitely touch on the stuff from the rest of the slide deck on Wednesday.
Thank you.
Yep. Any other questions, guys, about this stuff? If not, let's take a 10-minute break and we'll come back at let's take a 12-minute break and come back at 11:05. Okay? And then we'll discuss. If you have any questions between now and then on this material, feel free then, or we can just, because I don't think the journal club thing will take all 2 hours. So we can use that for some other stuff to get into. Okay? All right. I'll see you guys in a bit. Again, these Labsters are less than ideal. I would much prefer to be on campus and having us doing experiments face to face and growing up things. And unfortunately, that's not happening. And something has to be done for you guys to get the lab credit if you're transferring. If this micro class didn't have that lab credit on its description, then that'd be a different story. But we've got to do it, and we got to get through it, and that's why we're doing these readings, and that's why you're doing these Labsters, because I can't think of much else to make this work. So try your best to get through it. If you have an issue where you complete a certain amount and then it just crashes on you, let me know. I'll update it in the grade book. I don't want these labsters to be things that you just dread. And I don't want it to reflect on your grade if for whatever reason they crash. So they're not designed they're designed to help you learn. They're not designed for me to assess what you've learned, because again, none of this stuff is going to be on the test. I mean, it might overlap with material we cover, but I'm not going to test you specifically on these labsters. So that doesn't mean don't give it your best shot and try to do it, right? But I'm just saying, if you have issues, let me know and I'll correct it, okay? To the best of my ability. And that is just simply going into grade book and saying, oh, okay, you didn't get a score because you couldn't finish it. I'll give you a score.
I have a question or maybe suggestion. Can we possibly open a discussion forum for little glitches? Like one of the first labs we did, it took me, I don't know, 30, 40 minutes to figure out how to find a lab coat. And then in this last one, it took me at least half an hour to figure out how to put a new unused test tube into the tray.
Uh-huh.
And it just, you had to discard of one of the other ones first.
Yeah.
So,
Yeah. So I know that there are a couple different things we could do. One is that I could just pull up my Zoom on Wednesday. And then you guys, during lab time, you can chat about it. There's also something else that some students of mine used last quarter. And I'm blanking on what it's called right now, but it's got some strange name. And it's like an online forum thing where you guys can message each other, or you can post like the assignments that are in the class, not the answers, but like you can say, oh yeah, the professor assigned this, just so you know, or you can chat, and that might be a way that you guys can do that to where it's not dependent on a specific time for me with this Zoom. That might be an easier way. I gotta think of the name of that. Does anybody else have any recommendations? Some kind of like app or something that you guys can use?
I think you can just do an open discussion that doesn't close on Canvas.
Uh-huh. Okay.
Yeah, that would be easy. We've done it I do it in couple other classes, but
They do have a chat area as well on Canvas that's real time.
Okay. Yeah. Is that what you're talking about, Christine?
well, I think the chat, you have to actually be there and somebody else has to be there to chat. With opening just a discussion post on Canvas, you can post it, leave it there. When other people are on, they'll see it. But I think chat, the two people
I can still see the messages from days ago from chat.
Oh, you can? Okay,
Yes.
awesome.
Okay. So yeah, I mean, if you've had me before, hopefully you know that I really encourage you guys to work together and be, colleagues and co-workers on this endeavor of learning. And so I feel like you guys learn much better from each other than you do from me. And that's my philosophy on it. So
Ouch.
it's true. I mean, I feel like you're peers, right? And the information comes better when it's from peers. And so I think that, yeah, if you guys want to use the chat or the posting, like I didn't know that was an option. So by all means, I encourage you guys to do that. So does everyone know how to access that? Does anyone not know that would like to know? I'm assuming, I mean, I see the chat on the side. I'm assuming that's what you would do, but
Correct.
Okay.
Yeah, it's in Canvas and it's per class, so only the class gets to see that chat.
Perfect. Yeah, I really would love for you guys to try and utilize that. So let's see here. Let me share my screen. So yeah, if you look over here on chat, click on that, and anyone struggling to get on Zoom this morning? Well, that was from last week. So yeah, so Ryan's familiar with the chat. So yeah, use that, guys. That's a great way to get that interaction. And I encourage you guys to use it for your research proposals too, if you want, and so on. I mean, unfortunately, in a different world, you guys would have hung out in the science commons with all the preserved animals and discuss this stuff together. But we gotta make do. So
I had a question actually about the proposal. Was there a rubric behind that, or were we just pretty much sending you a couple sentences or a small paragraph?
Just for the topic, no rubric. Just give me an idea of what your focus is.
Cool, thanks.
Mm-hmm. Now in a couple weeks, not this week, but next week, I'll want an outline. And so again, it's up to you guys. It's up to you guys to figure out how much information you're going to have at that point. Okay. I'm not going to dock you because you don't fit a certain guideline. That's going to come when we get to the actual proposal that you turn in at the end. But this is just, meant to encourage you to move along with the project. And so it's up, you give me what you have. And if I feel like it's kind of on the insufficient end, you may not get all 10 points, but do your best. Okay. So with that in mind, we'll adjust Labster accordingly. I really recommend that you do it during the lab time, like I said, because that gives you a chance to, if you have hiccups, to chat with somebody or to email me and give me a heads up. If you're saying, oh, it's Sunday evening, I got to do this Labster and it's taking hours, then you're out of luck. So please try to do it, during that assigned time. And then the quiz. Okay, so the quiz didn't go as well as I was hoping. And part of that is because the auto grading just didn't work as well as it should. I don't know why. But then, and so some answers were left blank, and I don't know if you left them blank because you didn't know them or because it just, for whatever reason, didn't, accept your answer. And some of the students said, hey, I did the whole quiz and it's saying that I didn't do parts of the questions. And so, or I answered this correctly and it's marking it incorrectly, or I wrote one instead of first. And I tried to correct some of those things, but the ones that were blank, I don't know what to do with that. So this is what I'm going to offer. If you did well in the quiz, perfect. If you didn't do well on the quiz and that's because you just weren't prepared or because there were glitches, I'm going to give you a second opportunity to take the quiz again. All right. And let's shoot for that on Wednesday. And I'll figure out a time when to do it. Maybe we'll just do it right before lecture. Or maybe we'll do it. Well, what do you guys think? You want to do it right after? How about if we do it after Wednesday lecture? That way, that's technically lab period. That's lab period, and yeah, okay. So if you weren't happy with your score, I'm giving you a second option to take it again. I don't think I'll change it. Maybe I'll change one of the questions, just to make it slightly different. But it won't be out of line of that material. Okay, so the stuff that we went over Friday won't be on there if I do swap out one question for something else. Okay, does that sound fair? You guys okay with that?
Yes, thank you.
Okay. And if for whatever reason you didn't take the quiz at all, you have your one get out of jail free this time around, okay? But don't expect this to be a trend, okay? So we'll normalize, we'll get this quiz back in action. Oh, I see a little cat. Emily's cat. Is that your cat, Emily? Oh no, is that Rachel's cat? Or that's Christy? I don't know who's. I lose track of the animals here.
That's Rachel's cat.
Rachel's cat. Okay.
That's Chicken.
I have some gophers invading my backyard, and my neighbor just showed me a video of a coyote 2 mornings ago, got a mole out of his yard, like just dug out, pulled it out and yanked it around and killed it. And I have a fence, so like the coyotes can't get in. I mean, that was kind of the point, but now I'm wondering, too bad. But now my neighbors have these cats and they patrol our area because I have a sandbox in the backyard that I think they like to use as a litter box. But I put up with it because every once in a while they kill some underground rodent and display it in my driveway for me, like as, here, look what I did for you. So I don't mind the cats much, but then yesterday I had a cat come into the yard, and then my son got out this like gong, so bang, 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 and scared the cat away. And I was like, oh, no. Anyway, digress. I don't know why.
Chicken likes to bring live squirrels into the house at 3 a.m.
Well, see, I don't have any cats
So
myself.
fun.
Yeah, I know they do that, right? And my neighbor who has all these cats, she's like, I thank her occasionally. Oh, hey, your cat, I think got a mole for me. Thanks. Like, yeah, you don't want to see, it's a bloodbath in our house. What that cat brings in for us, proudly. So yeah, tributes to the sandbox god, exactly. So yes.
Professor?
Mhm.
Yes.
Hi.
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 Hi, Professor. Yeah, I have a question regarding to the quiz. I feel like too much writing is it takes a lot of time to write, and somehow I feel like very nervous to get out of the time. Instead of that, could you put more like matching or choose the correct or fill in the blank? So in that case, it's less time for typing too much time like that.
Yeah, I mean, I'm open. I'm happy, like I said, to get feedback from you guys and adapt accordingly. So the thing is, I try to design this to where you really, when you take the quiz, you have to know the questions, right? If I give you too much time, then my concern is that you can just go to the slide deck or your notes and look it up and then spit it out. That said, I'm happy to reevaluate if there's a consensus that there isn't enough time for the quiz, and maybe
Sorry, if you're worried about cheating, my A&P class does Proctorio, which actually it may be a little more complicated because you have to have a webcam and a microphone, but it records everything while you're taking quizzes and tests.
Yeah, and I mean, that's definitely an option. I tend to, my inclination is to resist Proctorio because I think it just seems like Big Brother-ish to me, and I don't know. We can go that route, or we can go the route of you have a limited period of time, and if you know the material, and if I can dial it in to where you have enough time but not too much time, that would be my preference. But if you guys would rather do Proctorio and have more like different questions or something, we can talk that through, right? You guys are the ultimate experiment right now for me because I've never taught micro in this way. So you're the guinea pigs, just like we'll talk about these poor patients that had that were part of the Tuskegee project. So I'm open to feedback. You guys let me know. We can talk about it now or offline. You can send me an email and say, or message on Canvas and say, hey, I really am struggling to have enough time to do these quizzes. I try to have most matching and fill in the blank. There's a couple essay. This one had more writing out than most quizzes. And if you guys feel like that was too much, that's good to know. But
Okay, maybe I try to speak up my typing.
Yeah.
Yeah. Thank you, Professor.
No, thank you for bringing that up, because if you don't tell me, I don't know, and I just assume that it's going great. Okay.
Yeah, thank you.
And I'm not someone who's so stuck in my ways that I'm not going to adjust things, because I want you all to succeed. I want you to learn, of course, and I have to assess that, but I want it to be fun. I don't want it to be something that I don't want you to think back on micro with negativity because it was unpleasant, right? So let me know, shoot me some messages. We'll see how the next quiz goes. I'm giving you an opportunity to retake this quiz from last week, and then be ready for this Friday. Okay?
Okay. Yeah, thank you, Professor.
Anything else before we jump into the journal club topic? All right. Who wants to set the stage for the group? What was this thing? What happened? What was it about? Remember, it's a group discussion. Brave soul. Just
Okay, I'm not shy.
there you go. Thank you, Christine.
Okay, so yeah, it was about looking at long-term effects in syphilis on African American men over what, like 4 decades, thereabouts. I think it started off a little differently with a different idea in mind than that. It actually seems like the original idea was to actually train doctors and nurses to in the lab and to be able to test people, and then also to actually give people treatment for syphilis. But then it seemed that the people who are funding this project were super worried at the high rate of infection among African Americans and that it would look bad. So they decided not to keep funding it. And then the experiment changed and it became not treating people for syphilis, even once an actual cure for some of the earlier stage of syphilis was found. And these people thought they were getting treatment for it, in a nutshell.
Yeah. Anybody want to add to that? And just the overview at this point. So syphilis is sorry, Hava.
I was going to add to it saying that it's interesting that the only reason why it even stopped this longitudinal study was because of the public outrage of someone reporting the study and what was happening.
Yeah, right. I mean, exactly. You don't change until you have to. Not out of conscience. So syphilis is a bacterial infection, and you can have primary symptoms, you can have the secondary stage, or you can get into the tertiary stage, which is the later life symptoms and signs. And a lot of historical figures, it's believed that they had succumbed to syphilis. I believe Christopher Columbus is one who they thought, based on some of his later on behaviors, that he might have had syphilis. And so, yeah, the Tuskegee Institute was set up with good intentions to empower the African American community, to teach and train African Americans to be able to be physicians and researchers. And not only, that benefits them, but it also benefits the community, especially in impoverished areas where a lot of African Americans never had even been to a physician. And maybe there was some mistrust on whether or not, you would go to a physician that was of a different race, and would you get the same kind of care, or would you be experimented on, like what happened in this project? And so you're right, Christine, it started off with, I think, good intentions and then morphed. The Tuskegee Institute was also involved in the first commercialized expansion of Henrietta Lacks's cells. So that guy in the basement who turned her cervical biopsy into immortalized cell lines then he was passing around to different researchers, but then they's like, okay, let's set up shop and let's really like crank these out. And they used the Tuskegee Institute to do that. So that's one of the parallels. Another parallel is that these diseases were considered bad blood. That's what it was called colloquially. And so I think in the excerpt from Henrietta Lacks, it mentioned that when she first had these symptoms and didn't know that it was cancer or anything, she just assumed it was bad blood from her husband being unfaithful and sleeping around on her. And so we're talking about a group that doesn't have any idea what an STD really is, and whether or not it's an infectious disease or it's a cancerous disease. And so with very little information, it's hard for them to seek the proper treatment. And so what happens is they get enrolled in this program and say, hey, look, we're going to pay for your meds, and we're going to if and when you die, we'll pay for your burial expenses. And as a result, we get to look at you, and like, for them, they're like, oh, okay. And so that right there was unethical. But then, I mean, what happened? What did they do that was really the worst part of this project?
Well, they did the testing on them and didn't actually treat them. They said that they were treating them up to a point and stopped, so they can still have the disease, and people still thought that they were being cured for it.
Yeah. Right. So they were treating them to where they weren't necessarily infectious, but they weren't cured. And why? Why did they want to do that?
Because they wanted to see the longitudinal effects of syphilis if it went untreated.
Yeah. So in Norway, I believe, in the early 1900s, they had done a similar study, and they found a lot of information, but particularly it was done on Caucasians. And so they were curious if African Americans or even Africans, because of the link with sickle cell disease and its associated lower rates of infection of malaria, they wanted to see if there was a different manifestation of disease in that. And so they allowed these patients to think that they were being treated, and all the time they weren't. They were being systematically monitored to see what it does in a longitudinal study, like Hava said. They wanted to see how it manifested over time. And right, I mean, that is, so when we talked about Henrietta Lacks, it was a little bit more, I think there was some wiggle room for the unethical nature of it, right? And we discussed that. Like her overall, her care, her as a patient wasn't affected by that at all, right? In fact, she got the standard of care at the time, and she still succumbed, right? And she succumbed rapidly to it. So what they didn't impact her from a patient treatment perspective. But this is very different, right? This very much impacted their projection and their health.
Can I add something to that?
Please, yeah, do.
I just went back into the article, and starting in 1933, they tried to get permission to perform an autopsy when the men died from the study.
Mm-hmm.
I think that means they were kind of playing the long game when it came to what they were looking to do.
Right. Yeah, I mean, even at that point, they were trying to figure out what this was doing physiologically by using humans as test subjects. We don't do that anymore. And in fact, in microbiology, oftentimes back in this era, grad students would volunteer to ingest some, microbe in order to see if that was really what was causing the disease. And there was all this pressure because, let's say you're a grad student who's working on your PhD and this is your project. Does this bacterium cause this disease? And you really got to have a human because it's not infecting mice. There is an unethical pressure for you to advance your own research by being a test subject. And so nowadays, I mean, we don't do that, but it wasn't unheard of back in the day. A lot of medical research that when we think about Nazi Germany, we think about often the medical experiments that took place on the Jewish community and how awful that was. And we think that, my gosh, how could people do that? And it's true. Yet in our own backyard, not to the same extent, like I'm not trying to compare the two, but what I want to say is that, during the 1900s, a lot of bad stuff happened, and it wasn't just Nazi Germany that was doing it. It was happening in our own country. It was happening in other places in Europe. And, by discussing this, it's good that we relearn the true history of things and how it played out and where we are now. So in looking at that assignment, some of the questions were, let's see, one question was, what do you think the motives for the people involved in the study were? Okay, so let's dig that apart a little bit. What's the motive of the patients? We kind of mentioned that, but why would the patients be interested in enrolling in this project?
Free healthcare. They reserved free meals when they went to checkups. They also get burial services, so their family gets money to help in their passing. There's like a lot of different aspects for them to be enrolled in it.
Yeah, these are impoverished areas. It's taking advantage of someone's socioeconomic status to get them to participate. Now, do we think that everyone involved in this study, the personnel and the staff and the physicians, the nurses, all of them, do we think that there were heinous ulterior motives? Were any of them potentially naive to the grander scheme? Did anyone think that this was benefiting the community?
Yeah, I think sorry. I'm sitting here with Emily, so our speakers are like, I think Nurse Rivers, I think that was maybe, like, if we were to not read into it, we could look at it and say, oh, maybe it was malicious because she like knowingly was not allowing the subjects to get treatment. But I think from her point, she got a job to work in the healthcare industry, working with physicians as a nurse, as a Black nurse. And she got to help her own community. She got to give back. And also she was just following orders from these physicians, and some of the physicians I believe were also Black. So we could go into that, and were they being malicious, or, was this just they didn't want to taint the study? But I personally think that, I think it was, what's the word, like, yeah, benign, or just not, it wasn't malicious. I don't think she was trying to stop them from having access.
She had altruistic motives and just wasn't aware of the details of the study at that level. So not everyone involved in this was part of the conspiracy.
I think it didn't it say that she did know, and most of them did know what was going on, but I think it made mention that well, before the penicillin and cure came about, the actual treatment could have pretty severe side effects, possibly even death. So I think it said that, you know, she thought, well, sometimes the treatment for this is pretty awful. So that was one thing. And then the other part for her, I think it said it was that most of these people didn't have access to healthcare at all. And they were getting treated for a lot of other things besides syphilis that they wouldn't have been otherwise. So maybe that played a part into it. And perhaps over time, she just got so used to what was happening, but that by the time the penicillin came about and a cure came about, she was just so used to and ingrained with the system.
Yeah, so if I get what you're saying, it's like maybe for a while the ends justified the overall means, and that there was downstream benefit, you know, even though this kind of darker part of it was going on, that some patients were benefiting in other ways, and that, you know, maybe she wasn't. Yeah. Okay.
I think my issue with that argument is that it wasn't, I don't think it should be anyone's choice other than the patient's for whether or not they want to take treatment, you know? And I don't know, I just feel like they were dehumanized. They wouldn't even call them patients, they would just call them subjects. So I don't know. That's pretty controversial, I guess.
It's tough. And, you know, one thing to keep in mind is that, you know, this was before HIPAA, and this was before a lot of psychological and sociological barriers were dismantled. And I don't think it gives anyone a free pass. You know, you can't say, well, it was a different time back then. That's inexcusable. And in fact, it does talk about people who had issue with where this project was heading, and then they, you know, subsequently were no longer a part of it, right? And so, yeah, I agree with you, Sarah. I think that it is heinous, and they were dehumanized, and to go along with that, even though it's not excusable, sometimes we have to factor in what the pulse was at the time, you know, and where people's minds were, and it doesn't take away their culpability. It helps us understand how something like that happens, so that we can hopefully not let it happen again, you know. And I think that things like this are beneficial because they are a study in overall human psychology and where we are at, and how we get to where we are now, and how we progress and get better in the future. You know, we are still struggling with issues in race, and maybe not from a medical or a research perspective, as far as I know, but we're still seeing the same grains of issues pop up, right? And so what can we do to move forward? And I think that's the big question, right? That everyone is, I don't know if you hear that, but one of my kids is in trouble, hopefully. But anyway, so yeah, any other thoughts? That's great. Great.
Well, yeah, just to add on to that, I mean, this was before the civil rights movement, and Black women and men obviously had less rights back then, but it's still not right, but it's the mindset that America was at the point, and most of the world. I mean, we've always had slaves. There's always been, you know, Native Americans, Africans, Irish, whoever, but it's it's history, you know.
Yep, exactly. So,
you know, I'm sorry. One thing that I was really surprised by when I just did a quick Google search of medical ethics cases, I was actually kind of blown away with what I found, and how recently some things have happened where I wouldn't have thought it could happen. in closer to this day and age. You know, a little further back, but closer to home is Willowbrook. That, you know, and then there was something I think involving the state of New York with infants and the consent, what is normally needed through parents, but these children were wards of the state. And how does an infant give consent anyway for an experimental procedure? Or things like, oh, let's move it to another country where the rules are really lax, and it's American pharmaceutical companies. You know, I was actually really surprised at what still happens more recently.
Yeah, I mean, it's a process, right? We don't just flip on the switch and say, oh, hey, what we've been doing is wrong, we're going to stop. You know, it trickles, and we get awareness to more and more things, but it's a slow process, clearly, right? And so, you know, along those lines, do you think something like this could happen today in the US with, and that last question, I think they used HIV, AIDS as an example, but are we beyond that now? Can we safely say that, at least in the US, that something like this would not happen again?
Aren't there reports recently about like immigrant women being sterilized?
Yeah. Yes. So the ICE, there were, I want to say it's in Florida, maybe. There was an OBGYN who, my understanding wasn't even board certified, was doing sterilization on these detainees.
Yeah, heard
so
about that.
yeah,
That's
and so
crazy.
I don't know much on this. My general thought was that it was kind of a rogue thing, that it wasn't it was this particular physician doing this. It wasn't and I don't know, but it doesn't seem like there was a group of people doing this for any reason other than just that one-off thing. But that may not be the case. That was it's kind of how I feel like. Anybody else have anything to add to that?
Yeah, so one of my friends doesn't live here. She's been trying to get pregnant with her partner for quite a while. She had a miscarriage last year, so they tried again, and then they ended up having another miscarriage. And apparently the doctor wrote on her chart with the first one that they don't think that she's able to take a pregnancy to full term. They didn't tell her. Like, they didn't tell her at all. So like the doctor didn't give her full disclosure, because they're like talking about potential like lawsuits and stuff because of like the whole process they went through. And like the doctor did not tell her the full truth because like they didn't have to disclose like the full diagnosis or whatever to her. So I think that things like this do still happen, because I have a friend who's going through like a legal battle in fertility dealing with this subject.
And the idea is that, well, if she had known that, she wouldn't have spent the thousands and thousands of dollars the second and third time to do that.
And she wouldn't have had to go through the pain of like having a miscarriage. Like, that's not just like, you know, like that's still, she still had like a child inside of her that she had to deal with, like that psychological and that physical pain of like giving birth to like a child. That she had known that beforehand, like that would have saved her a lot of time, money, and like pain.
Mm-hmm.
So I think that stuff does still kind of happen. So
I think it definitely does happen, especially towards women and minorities.
So sounds like what we think is that there are clearly cases, individual cases, where patients have had their information disclosed, or they've had information withheld, or in the case of this ICE, it was, so, you know, when you can't speak English, you have to have an interpreter. And so there's consent, and we talked about informed consent, right, last time. And so the sterilization thing is something that is, again, kind of an isolated situation. And that's just, yeah, let's acknowledge that probably across the country there are isolated situations where things are being done that are unethical. What about a collaborative effort to do something of this magnitude? I mean, conspiracy theories aside, right? Are we at a point now with the foundations that we have in place, having to have an internal review board to go over any kind of human trial you want to do? Is something like this, do you think, able to crop up again, where we have a collaborative effort of physicians and scientists and nurses and even politicians and all of that coming together to look at a particular race to see physiologically how they respond to something?
I don't know if this is recent enough, but the one I was talking about with the infants in New York happened in 2004, and it was they used Hispanic and Black infants, and they were testing safety and tolerance of AIDS medications. So there you have doctors, nurses, politicians, powers that be all in on this. Granted, I don't remember what it said for how long this was able to go on. I feel like once we catch wind of something like this, more people are apt to step forward and get it out that, hey, this is happening. And maybe it can't happen for 40 years with most everybody being okay with it.
Mm-hmm.
But I think as long as they think that they can get around the rules somehow, it will happen, because there's money to be made in drugs and methods and such, is what I'm thinking.
Yeah. I think the question becomes, as we have focus on certain things and there's a momentum, like for instance with Black Lives Matter, there's a momentum right now for that. If that momentum were to wane over time, could we revert back to certain mindsets or mindsets that don't go away? Like, like, it's not that everyone changes and there's a collective awareness, right? There's just the kind of some mindsets change, and hopefully that's enough to progress overall policy and the way we do things. But then, lurking underneath, there's the mindset that continues, but just doesn't have an active role. And can we get to the point where those mindsets begin to flourish again?
I don't think so, honestly. I think people, once they're set in their ways, they're set in their ways. I lived in Florida for a while. I have parents and relatives that are Southern. I don't agree with racism at all, but at the same time, it's very hard to change those people that have grown up that way,
Absolutely. So you think, Ray, that given time and relaxed regulations or looking the other way, that it is possible that at some point we could fall back and start doing studies on a certain race of people or a certain gender or a certain socioeconomic status, and that could come up again. That's what I'm getting from you.
I think my point is, yes, there's possibilities for us to eventually get past all of this, but at the same time, I think it's not going to happen for another few generations possibly, because we still have people that are voting and people in politics that are, or that were raised this way,
Yeah, and I mean, I think even things like reversion therapy, I don't know if it's been banned all across the US, but up until recently, that was still occurring, right? So this stuff takes time, right? You don't have change. What's that?
What's reversion therapy?
So it was this process to take homosexuals and change their sexual orientation. by giving them certain treatments.
Oh, that's terrible.
And I don't know what those treatments were, but up until recently, that was still being done, and it might still be done some places. So these things take time. These things can't just change overnight, unfortunately. And so I bring this up, and I wanted to do this, not, because I felt like it's important to have these discussions, because you can go on your Instagram and post, change your profile to something in solidarity or make a comment. But I feel like the best way to invoke change is to have discussions like this with people that we know, and not just be in our echo chamber and so thank you. I'm glad you guys, for those of you who participated. I think it's important to do this now more than ever. And like, I don't like to get political, like I said, with my classes, because I teach science. But I think that when it overlaps with things like that, then it's my obligation to conversion. Sorry, not reversion. Conversion. Yeah, that makes more sense than reversion. Thank you for that clarification. And Hava put a link if you wanted to read up on that. So I think these are important things, and I encourage you guys to continue to delve into topics like this, because the only way we get better is by learning and by discussing and by talking about these things, even if they're uncomfortable and they're not uplifting. They're important nonetheless, right?
And I think that's one of the issues is, the majority of America, not to be offensive, but the majority of America isn't as intelligent as we think we are. And the majority, I don't want to say the majority, but a lot of people, like, they don't go to college, they don't graduate high school, this and that, and they don't have the means to be able to identify the problems in our country, So I think us as a class, we're more adept to be able to have these discussions than the normal American.
Yeah, and those discussions can be painful, especially if you have a family that has different philosophical viewpoints, or friends. You come to a point where, if I have this conversation, I might damage my relationship with them, and I don't really know how to bridge that and deal with that situation. I don't know if there's a good answer for that, but definitely awareness. Like, my life has continued. There are things that open my eyes, and I thought that I was one way, but then realized that I was clueless to a lot of things going on, right? And so it's a process of self-learning, and then learning with our friends and our peers, and just trying to make it better, right? And I tend to be more optimistic. I tend to think that these kinds of particular studies won't happen again in this country, but I'm only doing that because I have to have hope, because I don't want to. I tend to be pessimistic and skeptical in other ways, and so I got to balance it out somehow. I think that we are beyond something like this, letting a group of people carry a disease long term to see what it does to them. But we'll see. Any other thoughts or comments on this topic, or this particular assignment?
Sorry, Carl, not to go back to the conversion therapy, but I put in the chat a website that actually shows a map of the U.S. where it is and is not legal for conversion therapy. And in California and the majority of the states where it's supposedly not legal, it's just not legal for minors. So technically, if they're of age, they're still able to do conversion therapy.
Okay. Thank you for sharing that, Natalie. So clearly it is still being practiced, in places. And I don't know, that's a whole different topic of discussion of whether or not as an adult if you can consent to certain treatments, right? I think that assisted suicide is something that is illegal in some places and mostly illegal in places. But if you're a consenting adult, where do you fall into your rights? And maybe we can talk about that some other time. I think that's an interesting conversation. What therapies should be illegal across the board, whether you're a consenting adult of your own mind or not, and what things should be okay, allowed, legal, I guess, in other cases. But I think that's opening up a big other topic, and let's not focus. Let's not get into that. So,
yeah, I think that would be more for law school students.
Yeah, but yeah, okay. Thank you for sharing that, guys. And any other comments or questions for this? Okay. All right. I will, I'll make an announcement when the quiz, with the parameters of the quiz, but let's just plan on having the quiz retake Wednesday after lecture. Okay. And lecture's at 10. So at 10:50, give you a period of time after that, maybe from 11 to 11:20, have that window open. Okay. All right. If there are any other questions, shoot me emails and give me your feedback, and we'll dig into viruses more on Wednesday. Okay. All right. Thank you, guys. Take care. I go.